So after we map the RNA-seq into the genome, there are algorithms that you can use to check the overall RNA-seq quality. So there are algorithms such as RSeQC, which are widely used. You probably don't need to do this if you are dealing with cell line experiment or mouse that's freshly sacrificed. You take out the tissue, isolate RNA. But if this is human tissue, archival tissue, or you're not so sure how long this RNA has been in storage, or maybe this is your first RNA-seq experiment, you might want to check the quality first. The very first QC measure is the overall mappability of the reads. Ideally, you want 50% of your reads to be mapped to the genome, and the higher the better. And you can also look at the quality score of your read relative to the location of the read. And again, the higher the better. So this is usually the beginning of the reads, the quality is better. And as you go longer and longer in the reads, the quality decreases. And so over the last decade, what sequencing machines such as Illumina was able to increase really is this part. Initially, they could only guarantee high quality in the first 30 base pair, but later on they could make it to 50, 100, and now even 150 at pretty high quality. And so if the overall sequencing quality is bad, that may not be the problem with your RNA sample. It could be the sequencing lane failed. If you see many other samples with overall low quality reads or quality decrease very quickly, supposedly you are sequencing 100 base pair, but even at 50, the quality score starts to drop, you might want to talk to your sequencing facility. There are some situations occasionally when the first few bases might have bad quality. And so you could just write a script to remove the first bases before you map them to the genome. So this is a figure to look at this is similar to our FASTQC, to look at across different locations, whether you have a fairly even ACGT distribution and you want to see a flat curve. It could be, depending on the genome, maybe A is slightly more abundant than GC in the genome, which is kind of expected for human genome, but this should be fairly flat across the different bases. If you see these type of position-specific AT biases, that's an indication you might want to trim the beginning of the read. For RSeQC, there are some RNA-specific QC measures, such as the insert size. If you are dealing with paired and read, you might want to look at, what is your insert size? Most of the time when people are isolating the RNA to put it on the sequencing machine, they sequence like 200 base pair reads. So you can see this is probably in the early days of 50 base pair on each side. The insert size is about 100. So you know that, okay, this is a pretty good one. In recent years, when people are sequencing, like a 150 base pair, occasionally you have to check. I think the insert size now is probably updated to calculate the beginning, because the two reads can really overlap with each other now. You read into the other read on the opposite strand. The next one is a very, very good QC measure, which is how many of those reads are mapped to the coding exons into the genome. This is just a histogram. This is not the percentage, but you can see here, most of the read are aligned to coding exons, then to 3' exon, 5' exon, and the red. There are always some little noise, which is kind of expected. And then also, if there are junctions, meaning that within the same read, it covers 2 exons, you can look at how many percentage of those are previously annotated exons versus the novel exon, or maybe at least half of it is right. And then say, supposedly, previously it was annotated that the first exon is linked to the second exon, but then you realize that there's a third exon, which also it can map to. And so that's partially novel. Completely novel would be, totally unknown junctions before. And so you would want a majority of your splice junctions to be known. Another very important QC measure, especially for archival tissues, is the TIN score, or the transcript integrity number on each transcript. This is calculated on every gene, and at the end, there is a median TIN score, which is kind of looking at the median TIN across all the transcript. And so in here, you can look at individual samples, med TIN score, which measures the RNA integrity at the sample level. You want this usually to be about 50%. This is just give you an idea. If you have a high quality sample, so in here, you can see these samples are pretty good. Their overall transcript, this is calculated across kind of an average gene from the beginning to the end. You can see kind of most of the time, the read really covers the whole gene. But if the RNA sample is degraded, remember that happens from the 5' end. You can see most of the read landed on 3' end. And so if the med TIN score is less than 50%, you might be still be able to read out the transcript from the 3' end, but it could be that some transcripts are already degraded, completely gone. You just don't know. And you have to be very careful in interpreting those results.